Hello, welcome to another screencast where we'll be discussing respiratory physiology. 2B, we'll talk about how specifically here oxygen is going to be transported in the blood. So how oxygen is transported, and be able to describe and explain here the molecular structure of hemoglobin, the abbreviation here HB, and its role in oxygen transport. I think we'll deal with carbon dioxide transport in a subsequent screencast, just to keep it short here. So in previous screencasts, we talked about external respiration, that is the exchange of oxygen gas from the alveolar spaces into the pulmonary capillaries, and then also this internal respiration, the exchange of oxygen from the systemic capillaries into the cells for respiration. But what I want to focus on here is how that oxygen is actually transported in the blood in this sort of systemic part of the circulation here. Right? So in previous screencasts, we've talked about erythrocytes and hemoglobin, but I just want to bring to light here this image from the cartoon, and that say that oxygen, 97% of it is transported as oxyhemoglobin. Right? And the coloring book little diagram here, this is the hemoglobin molecule, and A, these little circles are the supposed to be the oxygen molecules here bound to it. So we'll talk about that hemoglobin in just a second. So the majority of that oxygen transported as oxyhemoglobin, that is bound to the hemoglobin molecule. But then 3% of it is dissolved just in the plasma there. Okay. So we'll be looking at this equation here in just a second, where deoxyhemoglobin, this HHB, binds with molecular oxygen to form oxyhemoglobin, HbO2, and then this hydrogen ion here as well. So that's a little more technical than we need, but what you see over here again is that some of this oxygen will just dissolve into the plasma, right? This fluid here being the plasma, but the majority of that oxygen as it dissolves in the plasma then will also bind with hemoglobin molecules that are located inside the erythrocytes. Right? So in previous screencasts we looked at erythrocytes, and so here is a colored electron, the scanning electron microscope. Here are the erythrocytes in a light microscope. So this red blood cell right here, the erythrocytes, we have between 4 and 6 million red blood cells per cubic millimeter of blood here. Now within each red blood cell, you have approximately 250 million hemoglobin molecules. So this is the hemoglobin molecule. Right? And so this thing in each red blood cell times 250 million. Now, the hemoglobin molecule is 4 subunits here, and you see them in orange, these globular proteins. This orange, the green, the purple, and then this red. There are 2 alpha chains, the orange and the purple, and then 2 beta chains, here the green and the red. So you have these globular protein components of the hemoglobin, but then you also have these heme groups, which I want to detail down here. So the heme group consists of this porphyrin ring structure, so that's all of these nitrogens and carbons and these rings out here, and at the center of each porphyrin ring is an iron atom, right? And this iron atom here is Fe2+, and when it is oxidized, it binds oxygen and shifts to the Fe3+ state, and when you do this, it turns red. So again, 250 million of these hemoglobin molecules per red blood cell. Each hemoglobin molecule has 4 globular subunits, and each subunit then has a heme component to it that again is a porphyrin ring and this iron atom, so that each hemoglobin molecule, right, can bind 4 oxygen molecules. Right? And so when you have this deoxyhemoglobin here, then when you oxidize it, you end up getting HbO2 plus then a hydrogen atom here. So this is the way that the majority of oxygen is transported in the blood. I recommend clicking on that animation and watching this as well. So here's from the coloring book a little bit more about hemoglobin. Again, the hemoglobin molecule is two parts here. There's the two alpha peptide chains, two beta chains. Again, those are the globular subunits. And then each of those globular subunits has a heme associated with it, which again is a porphyrin ring and 4 iron atoms. Now, what I want to call your attention to is that the hemoglobin molecule, when the oxygen binds to it, it's actually relatively difficult to get oxygen to bind to that hemoglobin. But once it does, it ends up binding in this capacity called cooperativity, or cooperatively. Right? So cooperativity, right, or cooperatively. So if we see the heme here, and then, or this is the iron atom there, and then there's the iron atom here, and the iron atom's kind of hiding in there, and then it's really hiding out in this heme group up there. Okay? So the hemoglobin, when it is not binding oxygen, it's in this tensed state, and it's difficult to get oxygen to bind to the heme group, right? So again, there's the iron atom there, there's the iron atom there, there's the iron atom here, and then these are the binding sites. But what happens is that once one oxygen atom gets bound on there, and so here we see the oxygen binding to this heme group, to the iron atom there. This globular protein part changes its conformation, changes its shape just slightly, and makes it actually easier for the second, the third, and the fourth oxygens to bind. Right? And then it becomes this relaxed state here where once one oxygen binds very rapidly, very quickly, the other 3 oxygen molecules can bind as well. And the coloring book does a nice job of making this analogy to these oxygens trying to get into the boat of hemoglobin here. And it's tough for one to get in, but once that one guy gets in there, he can actually help pull the second, and those 2 can help pull the third, and then those 3 can help pull the fourth into the boat here. And then you have the hemoglobin molecule completely, as we say, now saturated. The hemoglobin molecule is saturated with oxygen molecules there. So the oxygen will bind cooperatively, and the reverse also is true. So when one of these oxygens gets off the hemoglobin, or is unloaded from the hemoglobin molecules, unloaded from the boat, the other 3 tend to dissociate from the hemoglobin actually pretty rapidly as well. So here we go. We'll take a look at this. What this looks like, kind of image-wise. So here's your red blood cell inside the capillary, and here's the alveolar air sacs here. Or again, to remind us that the PO2, the partial pressure of oxygen here, is about 105. As the blood enters this pulmonary capillary here, again the PO2 here is about 40. And so oxygen is going to diffuse down its concentration gradient, down its partial pressure gradient rather. And we talk about the loading of O2, the loading of O2 onto hemoglobin. And so we see we have deoxyhemoglobin plus oxygen gets us then oxyhemoglobin right here. And as this red blood cell moves past in this direction, the red blood cells, they line up one at a time, and as they pass through that pulmonary capillary, they actually get loaded with oxygen, and the oxygen is then transported thusly. What's not shown here, again, is the 3% of oxygen that just simply goes into the plasma and gets dissolved directly into the plasma. But again, that's only about 3%. Right? Most of that oxygen is transported as oxyhemoglobin. The reverse then happens when we get into a systemic capillary, as the as the blood's flowing now this direction. The PO2 inside the tissue cells here is rather low, right? So somewhere in the neighborhood of 30, and the PO2 here is about 105. Again, so if as it's leaving this pulmonary capillary, the PO2 would be about 105. And so what we have here is the unloading of oxygen, or the dissociation of O2 from hemoglobin. Right? And so we get this reverse reaction where the oxyhemoglobin makes the hemoglobin, deoxyhemoglobin, and the oxygen then is able to diffuse down its partial pressure gradient into the tissues there. And so then by the time this the blood leaves here, the PO2 is about 40. Right. And then we'll return back to the pulmonary capillaries to pick that up. Okay. So again, watching these animations is helpful, and I think that I will stop there, but now we can just take a look at this cartoon here where let's follow the oxygen. So here are the oxygen atoms, here are the circles, and you'll see oxygen going into the airways, oxygen here and here. Here we see the loading of oxygen onto hemoglobin, right? So we get this cooperative binding right there where one binds, and now this molecule right here is the oxyhemoglobin, HbO2. Coming into that pulmonary capillary was the deoxyhemoglobin right here, and you notice that that not all the oxygen is actually taken off of that that hemoglobin molecule. So there are still some bound there. The hemoglobin is not completely unsaturated there. We can talk about that later when I discuss the dissociation curves. Right? And so again, we see some of the oxygen is dissolved in the plasma here. As the hemoglobin molecule is saturated, it reaches the systemic capillaries. The partial pressure gradients help get that oxygen off and into the cells. Right? And then here's your mitochondrion over here gobbling up the oxygen in aerobic respiration. Right? This right here helps to keep the PO2 inside the cells here relatively low. Right? So then this deoxyhemoglobin here is then transported back to the the pulmonary capillaries to pick up more oxygen. Okay, so I think we'll stop there, and you can take a look. Actually, well, we'll just take a look at this big picture here. This is reversed of what we've been looking at, so I just want to point out that external respiration is at the bottom, internal respiration at the top of this diagram here, and so again, you'll be able to follow the pathway of oxygen being transported again from where it's a high PO2 here, right, bind in the alveoli, right, down here to then binding to the hemoglobin. So here's deoxyhemoglobin coming in, here's oxyhemoglobin going out of the pulmonary capillaries, and then this is pretty much our saturated hemoglobin molecule making its way up here to the tissues, right? And then here we see it dissociating right here. We see the dissociation of the oxygen from the hemoglobin. Then we get a deoxyhemoglobin here. Again, notice that it's not completely unsaturated there. There's a little bit of oxygen still left on there. Okay, so just pause here, and again, you should be able to now describe how oxygen is transported in the blood and describe how the molecular structure of hemoglobin is related to the transport of oxygen. Okay, thanks for listening, and watch the next one on carbon dioxide transport.